Welcome everybody to our July coffee hour. My name is Agnieszka Carpenter and I'm the Executive Director of BioME. BioME is Bioscience Association of Maine. We're a trade industry group devoted to advancing bioscience in the state of Maine and beyond. And Coffee Hour is our monthly series, a virtual event where we bring in 4 exciting speakers talking about new companies, new developments, research, and some support or services for the life sciences community in the state. And today we have a set of 4 fabulous speakers. Each of them will have about 10 minutes for a quick presentation. And then at the end, we'll have a 10 to 15 minute Q&A. A couple of logistics before we go. The chat function is on and we encourage all of our audience members to leave your questions there. And we will address those questions at the end. So please feel free to just leave them throughout the event. You can also just say hi or introduce yourselves if you would like. And before we start, I also wanted to thank our sponsors, because without these great organizations, we wouldn't be able to put together events like this. So thank you so much. And a quick plug-in, our next event is next month on August the 23rd. We're holding BioME Summer Networking Reception. It'll take place at the Southern Maine Community College, 5 to 7 p.m. We'll have great drinks, food, and ocean views. So if you're free that day, please feel free to register. The registration's open and we will paste the link to this event in the chat as well. And thank you so much to all of you for joining us today. I think we're ready for the first speaker, and our first speaker today is Jaimie Logan, Executive Director of the TechPlace. And Jaimie, whenever you're ready, you can share your screen. Jaimie, I think you may be muted.
Yeah, can everybody see the screen okay now?
Yes.
Great. Good morning. Good morning. Thank you, Agnieszka, for the invitation to join the coffee hour this morning. I'm delighted to be here with everyone else to share some information about TechPlace and our businesses here, and about the support, our support for the biotech businesses here and the biotech industry in general. Just a little background about let's see. Now, just want to make sure. There we go. Just a bit of background about TechPlace and kind of how we got to be here. TechPlace is a technology science manufacturing incubator. I work for MIRA, the Mid-Coast Regional Redevelopment Authority, which was charged with redeveloping the former Brunswick Naval Air Station, which closed in 2005. And as part of the redevelopment efforts, they discovered that the landing in which the Naval Air Station became known as Brunswick Landing, they were able to accommodate very large businesses and properties, but they didn't have space for the small early stage and startup companies that needed that size and scale in order to begin their operations. And there really wasn't anything else in the general area as well. So MIRA took an existing facility here. It's about a 95,000 square foot building with a hangar attached to it that was a maintenance facility for aircraft, and turned it into a shared manufacturing and co-working space for tech companies. So next slide. So our focus is on clean tech, aviation and aerospace, IT, composites, and biotech. And this is a TechPlace is kind of an all-in-one. It's a very unique space because it offers so many different things for people. We have office space, we have shared office space, we have manufacturing space, we have shared manufacturing space. We have all the comforts of home with a kitchen and a game room. We have conference facilities with full video conference capacity, and mailroom. It's all inclusive. Your all your expenses are included. Your Wi-Fi, your utilities. It's all there. So it's great for an early stage or a startup company because you know what your expenses are going to be. We Next slide. So here's a couple of slides just to show some of our office space, our game room. And also we have one of the most attractive features about TechPlace is its ability to offer prototyping space for our companies. One of their biggest obstacles for a startup or a young company is getting enough money in general, but also to buy equipment or purchase access to equipment that they need for their R&D and their prototyping. And we have that here at TechPlace. We have our TechWorks facility, which has a wood shop, machine shop, and welding booth, and these are all shared, as well as we have a full composite layup facility and a biotech laboratory. And I apologize, these photos are kind of not current. This is when this was brand spanking new. It's loaded up with equipment and people and things now, but I need to get some new photography. Just a little bit of the equipment that we offer is on the screen there for you to see. Biosafety cabinets, incubators, centrifuges, a lot of freezers, fume hoods, microscopes, autoclaves, pretty much, and it's set up so we have shared space with bench space, 6-foot benches, as well as 2 private labs. So people can come in and just rent a bench space and be able to access all the shared equipment, and they can bring in some of their own equipment as well, which is terrific. We partnered with MTI to get the funding to buy the equipment in the facility. And we have lab managers, Dr. Jem Gire and Veronica Acorn of Salmonix, a company you'll see in a minute, who oversee the lab and onboard our users. But we find the best way to tell the story about TechPlace and our businesses is really to talk about the businesses themselves, how they've started here and how they've grown. Christine Logan, no relation to me, started TechPlace, and she is now the head of MIRA, the executive director. But she said when they first started TechPlace, they really weren't sure what kind of mix of companies they were going to get. They were thinking they were going to get a whole lot of millennials developing apps. But what they found is that we really have a great mix of ages. Sure, we have some millennials and we have some mid-phase career folks, but we also have several people located in the latter part of their careers. They've been in industry, found out where the pain points are, and they feel that they're in a position now to solve problems, and they're working on things that they really want to focus on. So it's great. And not all these companies are from Maine. We attract, a lot of these people start up in their garages or their homes or the basements or their friends' facilities, but we pull people from out of state. We have people currently here from Connecticut, Chicago, several from Massachusetts, and we also pull from out of the country. We've had folks from Switzerland. We have people from Italy right now, as well as Australia. Excuse me. So I'll share a little bit about some of our users. On your screen now is Field Phytonutrients. They are a company that came to us out of Connecticut. This is kind of a fun one for me because in my prior career, I worked for the State Department of Economic and Community Development, and I worked with several of the businesses there while I was at DECD and helped them find TechPlace. So they're here, and it's great to be working with them. Field Phyto, they process algae to make an enzyme that goes into cat, dog, and fish products. They've commercialized the product, it's in distribution, and I think they're looking to get this into human products as well. Next up, Salmonix. Salmonix, and you may have seen Jem and company in the news recently. They won an award from the Manufacturers Association of Maine this past spring. Jem is also is our lab manager. They have an innovative business. They take a waste product from salmon farming, and they make a sustainable resource that is used to make high quality reagents that has a variety of health applications. So the photo that you see there that was a very rough manufacturing, just a bare manufacturing space, and they made a great little lab. That's one of their own private labs. Next up is Loam Bio. Formerly, you may have known them priorly as Soil Carbon. They come to us from Australia. Their headquarters in the US is in Minnesota. And they're looking, their efforts are related to development and launch of microbial seed treatments used by farmers to enhance their carbon sequestration. They are really taking off and ramping up. They are going to be, at some point, they'll be one of our success stories. They will outgrow our space within probably the next year. Another bio for us is Hound Bio. This is our newest bio company. They were founded in 2021, incorporated in January of this year. They now already have 2 full-time equivalents. And Hound Bio is working with immune modulating therapeutics directed to cancers. And this is pretty innovative stuff. It's reprogramming cells into cancer fighting cells. And that initial application is going to be for dogs and cats. The hope is that will find an application for humans as well. New England Oceanographic Laboratory, they do look at ocean sciences. They're studying the past, present, and future state of the oceans, the conditions, how they're changing, and how those changes impact the environment. And slide here, last slide, this Inspiro is no longer here. They left this spring, but they came to us from Switzerland, a biotech firm, and they were instrumental in developing our biolab. They had a private lab space, and they helped us fit out the lab space in general, the shared lab space. So what ties everybody together here at TechPlace is our community. It is part of being here is not just having space here, but interacting with each other. We have a lot of shared spaces, activities, social, educational, to kind of create those collisions of innovation we all like to talk about. They can talk to each other about supply chain, financing, where they find employees, just moan and groan and find some support with each other. And they have a lot of fun. It's a fun place to work. It's a cool place to work. We have a lot of tours throughout the area, and everyone comments on the community, and the members appreciate it, and they have great pride in showing everyone what they're doing here. So I want to just take one last moment to talk about the future of life sciences here at Brunswick Landing. We found out there is a real need for life science facilities here. We've identified that need. We're turning away people because we only have so much room. We can only accommodate so many people. We have some shared lab space and some private lab space, and that's it. So it's outgrown our capacity. Christine, the last few years, worked with Consigli and SMRT to design a 45,000 square foot facility. We've got the plans, we've got the location, and we are looking for a partner or an operator with expertise in the biotech area to do that operation. It's where we think it's going to be very successful, and it's going to meet a huge need. So I'm just going to give you a quick shot. This is not a final rendering. It may change. You may have a second story on it, but we are really putting out to you, the BioME community. Like I said, really just waiting for a partner. We're flexible. We can build it and you can operate it, or you can build it and operate it. We just don't have the bandwidth within our own team to take on that project. So it's going to have individual lab space, of course, shared lab space with equipment that can be used by everybody, office space, meeting space. It's going to be state of the art. And we're pretty excited about it. So the need is there. We have the location, the infrastructure, and the plans. We're flexible. And we think it's going to be successful. And we hope maybe you can help us find somebody to operate it. And last from our team to you, thank you very much for your interest in TechPlace. I'd be happy to answer any questions at the end of the program. Anyone like a tour of TechPlace, please just let me know. I'd be happy to show it to you. And thank you very, very much.
Thank you so much, Jaimie. That was a great presentation. It was, I especially appreciate that you introduced some of the companies in the TechPlace, especially the newer ones that our community might not be familiar with yet. And if anybody is interested in a tour, I highly recommend it because it's great to see it, actually go there and see it. And Jaimie and I are already planning a BioME tour of the TechPlace probably next year. So stay tuned and we'll share more details as they come. And now our second speaker today is Dr. José Fernández Robledo from Bigelow Laboratory for Ocean Sciences. And Jose, whenever you're ready. Yep, and you need to unmute. Jose, we still can't hear you.
All right, I think it's okay now, right?
Yes, thank you.
Well, thanks a lot for giving me the opportunity to talk about what I'm doing here at Bigelow Laboratory. Usually when I go to the BioME meetings, I'm the oyster guy. So sure, you're gonna be hearing about oysters. If I can move this. There you go. So oysters are ecosystem builders because they are filter feeders. There are, the oyster mittens is telling us that oysters are also a natural resource. However, now the population is much larger than when the natives were in this part of the United States. So because the oysters are very popular culinary and touristic item. This is very good for the oyster aquaculture. The oyster releases are increasing. This is in Dodge Pond. I used to hike there when I came to Maine 10 years ago, and there was nothing, and now I have this beautiful oyster release. Also, they are developing new technologies that allow them to work with the oyster baskets in a much more efficient manner. The way I describe myself, I'm a molecular parasitologist and a genetic engineer. So oysters and bivalves in general, they can also get sick, and they can, the agent that causes the disease are like for humans. You have viruses, you have bacteria, a large number of protozoan parasites, and also even cancer. Because they're filter feeders, the oyster reflect the quality of the water where they are growing, because they can concentrate, again, viruses, Vibrio coliform, even protozoan parasite derived from cows and cats. Of course, we are all familiar with the red tides, where the bivalves concentrate the toxins, and essentially you have to shut down the operation because you cannot sell it. And like any other live organism, they are subjected to all these environmental challenges. I do work mostly with Dermo and MSX. These are two parasites that are relevant for the oyster aquaculture. This is the last survey that we did in 2016 and 17. So the take-home message is that both of them are present in Maine. They can reach very high prevalence. The good thing is during the last 10 years, we haven't had any mass mortality. So it looks like the oyster aquaculture is able to survive or thrive even in the presence of parasites. And parasites are here to stay. It's very difficult to get rid of them. So I'm particularly interested in Parkinson's marinus. We call it dermo or dermo disease. This is a very clever parasite because it uses the blood cells in the oyster, the hemocytes, essentially to enter, propagate, and distribute into the body of the oyster. So the way I usually see it is like a leukemia, because these are the defense cells, and the parasite is destroying us over time. However, this is a very, very good parasite because we can grow it in the lab without having the host. We can grow it like you grow bacteria and yeast. You can reach densities of 10 to the 8th per ml. So this is very, very unique. During the last 10 years, I've been working on developing Dermo, the oyster parasites, as a new vaccine delivery system. So we already have shown that when you feed mice with the oyster parasite, it doesn't cause any pathology, but is able to engage the immune system. So we may have already a very good adjuvant. We developed the methodology to being able to deliver genes inside the oyster parasite. And then right now we are working in expressing different candidate genes for vaccines. And I'm going to be talking about this one. Also, to speed up the expression of the genes, we just published last year the CRISPR-Cas9 system for this particular parasite. So this is one of the genes that we are expressing, the gene that codes for the spike protein of SARS-CoV-2. Here, essentially, what we have is a plasmid that has the gene tagged with GFP, and you can see that it's expressed. When you use antibodies against the spike protein, they are recognized. So it looks like we have a system that can not only express the gene, but the antibodies can recognize the protein. As I mentioned before, in order to speed up the expression of genes of interest, we developed the CRISPR-Cas9. And where within in the lab, we have a mutant that is fluorescent all the time and expresses a gene targeted to the surface of the cell. So essentially, we were able to replicate the genotype, essentially using CRISPR. Gonna be changing gears a little bit. Clams, they can develop a cancer, and it's a contagious cancer. So it goes from one individual to another. So these are the blood cells. When you put them in a slide, they spread, and these are the cancer cells. And this is a Michael Medford. He still likes the clams, so it doesn't affect humans. You can keep eating them. And at the time, he was working at the HIV lab in Columbia University, and they were interested in this particular cancer that we know already for the last 30 years because the etiological agent is thought to be a virus. So essentially, he started studying clams, and essentially he wanted to genotype the cancer in the cell, in the clams. So he will. This is a normal clam. This is a clam that has a cancer, and he did that for a lot of clams. So when you have a cancer, your cancer cell should match your genotype. So what happened in this case, it will not match the origin of the clam, but it will match to each other. The genotype was identical. So this was telling us that essentially the cancer cell were jumping from one clam to another. So something very unique in nature. There is only two more cases. One is the Tasmanian devil with the mouse cancer, and the other is a STD in dogs. So at Bigelow, we have this program called REU. It's called Research Experience for Undergrad. This is Tejas and Maria Jose. They worked in this project last year. And his work was reflected in this paper that is already published, where essentially we show that using qPCR in environmental water, we are able to detect the cells. We also show in the same paper that when the temperature is low, the viability of the cancer cell lasts for weeks. So you have floating in the water for weeks, the cell that you're gonna go from one clam to another. And fresh water essentially decreases the viability of the cells. Changing gears again, we are working with oysters. We have a very, very nice genome of the oyster, and the number of predicted genes is 32,000. For reference, we humans have only 20, I guess, because we don't have to make a shell. So if you look at the function of the genes, you know, from these 32,000, essentially, we also have no idea. So what I've been doing, and also other group, is developing the tools that will allow us to put functions to the genes. So one of them is to be able to label genes, in this case with GFP. So we succeed transfecting Chrysostera virginica blood cells. And the way we use it is using one compound called dendrimers and a plasmid that is driven by a viral promoter. So using this, putting these things together, the compound makes goes inside the cell. And then if you have expression, you see green color. So this is one of the tools. The other tool is to develop cell lines. This is Shindy. She's working in the lab right now. We're trying to establish cell lines from Chrysostera virginica. Myself and a lot of labs, they have tried that you know, for years. Nobody has succeeded. So what we're doing different this year is we are using different tissue explants, but we are trying different media. We are also coating the wells with different compounds, and also we are using different substrates. And the hope is that some of the cells derived from the tissue plants will start propagating. So we're checking every day. In the oyster, one of the main challenges that we have is because they live in the water, contamination of the culture medium is always one of the big issues. And essentially, we have a oyster genetic system. We will be able to address the function of those 32,000 genes, but also some very interesting things that we have in the oyster. For example, I don't know, but if you use an extract of the shell, it induces bone formation, so it can be used in biomaterials. Oysters they don't have antibodies, but they have to fight with the same sort of pathogens that we do. How they do it, we don't know. So the microbiome, this is also very interesting because they live in the water, and the water is full of different microorganisms. So how they are selecting one particular one to be in their body versus another one. So it's a good system to do that. The biomass, they have antimicrobial peptides. We also think that by having our oyster genetic system, we can guide the hatcheries in the selective breeding program that they have. You know, we can study toxicology, we can isolate virus, and we can also learn more about, you know, the cancer in the clams. Just a reminder, if you happen to be around tomorrow, we have finally our annual open house after, you know, a hiatus of 2 years. And also we have started the cafe size where, you know, a scientist give a talk to the public, usually in terms that the public understand, and usually it's about a subject of the different research that we are doing at Bigelow. And that will be for today. Thank you very much.
Thanks so much, Jose. Oysters are always so fascinating when you talk about them. I'm so glad you said we can eat clams, because after last time you talked to me about oysters, I didn't eat them for a while, but I am eating them now. And if anybody came to the Women in Bioscience last month, there was proof of that. And Jose was eating them too, so they're fine.
I forgot to say that I still love oyster and clams, and this doesn't stop me from eating them. So eat oysters.
Great, thank you so much. Okay, so switching gears, we are moving to our next speaker, and our next speaker today is Dr. Frédéric Bonnet from Mount Desert Island Biological Lab.
Hi, hello everyone. So I'm Frédéric Bonnet. I'm managing the light microscopy facility at the MDI BL. And so today I will talk about what is available to you. So first of all, if you are not aware about what is MDI BL, so it's Mount Desert Island Biological Laboratory. So it's a non-profit research institute based in Bar Harbor. And our research is focused on regeneration and aging. So to support the research at the institute, we have a microscopy facility, and so I'm managing the microscopy facility. So the facility basically provides access to a state-of-the-art microscope and workstation, but also training. So if you want to use our microscope, you will be trained to use a microscope and also to understand optics. So the facility works as an imaging service. So usually it helps researchers during all the research projects that require imaging. So basically from planning to publication. So the facility is really implicated in research projects because the goal of the facility is to make sure our users are successful in their research. So on the facility, we cover a lot of applications. So basically we do 2D imaging, 3D imaging. We also have a software to do a 3D reconstruction and 3D analysis. And we can also perform a time-lapse experiment. What is unique at the MDI-BL, it's the range of sample. So we work with big sample, like axolotl to study limb regeneration, for example. So we have microscope to image this kind of big sample. And on the opposite side of the spectrum, we have some research lab who work on really a small sample like C. elegans, and they try to look at structure which are the subcellular resolution. So we have microscope to do a super resolution. So in total, the facility has 7 microscopes. So we have 2 stereoscopes to image full animal. So this is mainly to do a screening or a phenotyping. We have 2 wide field microscopes, one inverted and one upright microscope. So they are used for thin sample. So this one, it's mainly dedicated for cell culture and live cell imaging and color imaging if you want to analyze tissue, for example. This one, it's to do a 3D reconstruction or a thin sample. And if you need more resolution or if you image bigger sample, we have a confocal microscope. So we have here 2 laser scanning confocal, so an Olympus FluoView 1000 and a Zeiss LSM 980 with Eriscan 2. So this one is used to do super resolution. And finally, we have a Nikon, which is a 2 confocal in 1. So it's a spinning disc and a laser scanning. So the spinning disc here would be used to do a fast imaging. And if you want resolution, you will use a C2, so the point scanning microscope. So the facility is open access, so you have access to this system. And just to let you know, in the future, we plan to expand a little bit to introduce new technique. And this is tissue clearing. So if you are using big sample and you don't want to cut them, but you want to analyze them in the full size, so a full brain, a full animal, or a full organ, we will be able to provide a service for tissue clearing. So basically, if you don't know what is tissue clearing, it consists to make your sample transparent to be able to image deep and to don't cut the sample. And so this is a technique which prepares the sample, and to image this kind of big sample, we will build a light sheet microscope, which will allow you to image this big sample really quick. So we talk about a full mouse brain in less than 10 minutes. And so basically you can get this kind of images, so a full brain at high resolution. So this microscope will be custom made, so it's not commercially available. And so it's a tool we will provide for you if you want to use the facility. So this is an example of a mouse brain, full resolution. And you can also do some, another example of application, single neuron tracing. So you can track your neuron in the brain or along the full body of your axolotl or zebrafish or mouse embryo based on your question. So as I said, we are localized in Bar Harbor, but maybe you are far away, and so it's not easy for you to come and use a microscope. So we have this remote service. So basically, this service allows you to send sample to the facility. Then we will remotely image them. So basically, the facility will image, and we will do a share screen. We will share the screen like this. You can participate to the acquisition, and then we will send you back the full data set. And so basically, if you want to use this service, you just have to send me an email, and then we will do some tests to make sure the sample can be sent properly and not get damaged. And then we have to do some optimization. But if everything works well, basically in routine, you can use our facility from your place. If you have questions about this, you can contact me. And if you want more information about the facility, you can use the website. Thank you.
Thank you so much, Frédéric. It's great to see how MDI-BL is opening up and offering all kinds of services now. I didn't know about this, and I think a lot of our audience members haven't either. Also, just a reminder that this event is being recorded and it will be available on our YouTube channel after. So if you know anybody who might be interested in either MDI-BL microscope imaging services, or maybe interested in TechPlace, or would like to know what Jose is doing at Bigelow, feel free to share a link. We will also, per request, be sharing all of our speakers' email addresses if you would like to get connected after. Okay, so it is time for our last speaker today, and our last speaker is Louis Waterhouse from Law Calibration. If you have pipettes in Maine, you probably already know Louis, but we just wanted to highlight his work as well.
There we go.
Perfect.
All right. Hi, I'm Louis. Presenting's a lot of pressure for me. I wanted to gauge the room's sense of humor before we got too far into this. Law calibration is capable of calibrating pressure and vacuum instruments up to 20,000 PSI. Gravity of this presentation was really weighing on me. So wanted to let everyone know that we are an OHA certified distributor and repair center. We can calibrate anything from 6-place balances all the way up to 5,000 plus pound platform scales. Calibration is a hot market right now. We're capable of calibrating temperature devices thermodynamically from -90 to 1210 C. So that would cover anybody's furnaces or cryo chambers and everywhere's in between. Law calibration is something to write home about. We cover most every portion of electronics and electrical equipment. You know, are these puns kiloing you yet? And we, of course, think they're mega funny. Any questions? Hearing none, I'll just continue on talking then. So as you can see, we're very wide. We have a very wide scope of services that we provide. Anishka was kind enough to mention pipettes. We do have a full pipette calibration service, which we are accredited to. We cover torque, force, tension, linear dimensional. We can calibrate gauge blocks, and pretty much anything else in between. So we're as dynamic as all of the industries in this state, from construction down to biotech. There is not much that we can't do or can't touch, and can take care of all your calibration needs. We currently have a staff of 6 full-time technicians, all of which are very capable, very knowledgeable women, which is something that we're very proud of. The calibration industry is 93% male dominated. And so to have an all-woman technician crew is something that, you know, we are exceptionally proud of, and would love to see more women get involved in this work or in STEM in general. Yeah, I don't have any fancy pictures or anything. I just have that. But I'm free to answer any questions that anybody has. I think Agnieszka said that our contact information would be shared with anybody. If you know anyone that's looking for calibration work, we cover the entire state and most of New England. And we'd look forward to hearing back from anybody. And
Thanks so much, Louis.
I donate the rest of my time.
I just needed you to say something else too. So part of the reason why I wanted Louis to be here was because we are gearing up to our November event that we do at middle schools. It's called Maine Bioscience Day. And that's where we send scientists, biotech professionals to middle schools to talk about their work and to bring a hands-on activity sometimes. And we're looking for volunteers right now. And Louis has done it a couple times. So I don't know, Louis, do you want to share your experience a little bit? Maybe just say a couple sentences about what it was like to go to your local school.
It's an incredible opportunity. If you or your organization are not involved in it, I would encourage you to get involved in it. Seeing the looks on all of these children's faces is really a phenomenal experience. We've done it a couple times. We loved it. All of our people loved it. Just to be able to open up their eyes to the opportunities that are out there. Some of us that are natives and have lived here our whole lives, we've all heard of the brain drain or people saying, what is there in Maine to do? I can tell you firsthand from all the customers that we visit that, Maine is an incredible place. There are so many opportunities out there. This is a great opportunity to get your industry, expose these kids to your industry, knowing that, in a few years, they're going to be the ones looking for positions. They're going to be the ones that, will hopefully be changing the world. And you never know, you could be the spark that makes that happen. So I would 100% encourage everyone to get involved in that.
Perfect. Thank you so much. That was a great ad. And hopefully at least one person on the call will be interested. Louis, you can share, stop sharing. Thank you so much. And Jose actually did Maine Bioscience Day too, right, Jose? Unmute, please.
Yeah. It was one. It was very, very fun. The students were withdrawing blood from oyster, something that they didn't know they can do. Yeah, it was awesome.
Yeah, I remember some photos of the students with the oysters and being so smiley. It was probably the most smiley face from Maine Bioscience Day ever. I mean, I would be too if I were a middle schooler, and that's what I got to do in my science class. Okay, so we are now moving to our Q&A. I'm trying to sift through some questions in the chat here. Let me ask the first one. It's going to be to Jaimie. That was the first question that came. If you can just bear with me 1 second. Okay, so does the TechPlace lab space have air exchange exhaust for work in the lab space to accommodate light use of solvents and other common volatile lab reagents?
I believe it does. Yeah. You've got air handlers in there.
Perfect, thank you so much. Okay, there was a question about educational programs. So I wanted maybe to give both Frédéric and Jose a minute to talk about these. What kind of education programming do you offer for high school and college students? And maybe Jose, do you want to start?
Yeah, we have this REU, research experience for undergrad. Usually we get every year around 200 applications. So if you are lucky and you get selected, you will spend 10 weeks, with heavy work in the lab. It's very, very successful program. Then for middle school, we have this Bloom program. Essentially, it's a program where we get a high school student, one from each of the counties in Maine, and they spend 1 week essentially working with, we call it orders of magnitude. So they go from viruses all the way to large organisms. And we also offer training for science teachers. They can spend 1 week here learning about oceanography and marine sciences. At Bigelow also we have several centers. For example, the NCMA is where they have a collection of microorganisms, and they organize courses of 1 or 2 weeks. And I think, you know, if you want to get more information about those courses, the webpage at Bigelow Lab has much more information.
Okay, perfect. Thank you so much. And Frédéric? Oh, if you could just unmute, please.
Yep, sorry. Yes, so we have a program for students too. So like José says, RAU, and we have RAU and INBRI fellowship, which allow you undergraduate students to come during the summer, during several weeks, and they will be dispatched in the different research lab. And then we have also courses hosted on the institute. Some courses are funded by INBRI, so because we are INBRI Institute, like a lot of institutes in Maine. Anyone with an INBRI Institute can basically apply to these courses and come on campus during 1 week to follow the course. This year, we started the Maine microscopy course. So basically, it's a course I lead with Carissa Tilbury from Maine. And so we train people during 1 week about microscopy. So there is plenty of opportunity to learn, basically. So you can check the website or contact Jen Disney. I put the email in the chat, so to have more information.
Perfect. Thank you so much. And it's great to see that our research institutes have these types of offerings. Sometimes our community doesn't even know that these exist. Okay, and there's a question for Jaimie on the new lab space that's in the works. How has or will the project be funded? What would be expected? What would be the expected time from selecting a partner to being able to accept companies in, if you have that information?
Christine, just in general, Christine Logan would be the primary contact for questions on the Life Science Center, but I'd be happy to connect you with her. But we'd be looking probably at the EDA, state partners, private investment, kind of a multiple level capital stack. The EDA only if it's a facility that Mira would continue to own. Timing wise, we have we're ready to have conversations now. We have the plans with the design and construction already in hand. So we'd probably say about 2 years for completion. Now, you know, caution that or proviso that with, you know, how it is for materials and any backup and supply. But I think generally speaking, it'd be about 2 years.
Great. Thank you so much. Does anybody else have any questions? If you do, you can also just ask a question in person. No. Okay, so just before I start, I know that a lot of people joined later, so I wanted to show this slide again. We are organizing our August BioME networking reception on August the 23rd. It's going to take place at Southern Maine Community College. We will have great drinks, food, and ocean views. So if you haven't registered for that yet, the registration is open on our website, and we highly encourage you to join us that day because it's just great to see everybody at in-person events again. And other than that, I wanted to thank all of our speakers today, sponsors, and also all of our audience members. I was a little bit worried about a coffee hour in the middle of July, but everybody showed up, so that's great. And once again, if you're interested in connecting with any of our speakers, just shoot us an email, or some of the speakers have already been putting some email addresses in the chat, but we'll be happy to share with you. And I wanted to wish everybody a wonderful end of the summer, and hopefully I'll see you in August. Thanks so much. Bye, everyone.
Bye-bye. Thank you.
Thanks.